Cell cycle and cell division. So question number 1, growth of multicellular organisms is initiated by. The given options are A, cell division, B, cell enlargement, C, cell differentiation, and D, morphogenesis. So here the given options, cell division, that in a multicellular organisms the cells will divide, or if you take any living organism, the life of an living organism will start with single cell called as zygote, and in the higher plants the zygote will undergo mitotic divisions to form into embryo, and here the embryo is a multicellular diploid structure where the further the cells will divide and differentiate to form into an plantlet or an organism. So here initiation of the growth, that is starting of the growth. Initiation of the growth will start from the zygote. So that means first and foremost thing that should occur in a multicellular organism for initiation of growth will be the cell division process. Then only the number of cells will be added. It leads to the growth and development of the organisms. So from the given options, option A is the correct answer. Question number 2, cells in this stage can enter into G0 stage. The given options are A, S phase, B, G2 phase, C, G1 phase, and D, M phase. So here if you take the cell cycle, in this one the interphase and the division phase will be present. Interphase is a stage where the cell prepare itself for the next division process. In this interphase, G1 phase, S phase and G2 phase will be there. So here when a cell completes its cell cycle, it will further enter into the next cell cycle, where the first stage of next cell cycle will be the G1 phase. So here sometimes what happens, the cells will become quiescent and they enter into a resting like stage. So that resting stage we can call it as an G0 phase. Of course metabolically it is active, but it is considered to be the resting one. So after G1 phase it will enter into the G0 phase. So here from the given options, option C is the correct answer. Question number 3, mitotic division is also called equational division because the given options, A, chromosome number in daughter cells is same, B, chromosome number in daughter cells and parental cells is same, C, amount of DNA is same in both the daughter cells, D, amount of DNA is same in both daughter and parental cell. So here mitotic division or mitosis we can call. So mitosis is a division which occur in haploid cells and in diploid cells also. So here it is otherwise called as equational division. So it is why it is so called as equational division means, so here there will be no change in the chromosome number from parental cell to the daughter cells. For example, if a parent cell have chromosome number as 12, when this parent cell will divide, it will form 2 daughter cells, and each daughter cell will have chromosome number as 12. This one daughter cell will have 12 chromosome number and other will also have the 12 chromosome number. That is, the chromosome number in both the daughter cells will be equal, and at the same time the chromosome number of each daughter cell is equal to the parental cell. So as a chromosome number is maintained is same from the parental cell to the daughter cell, and daughter cells also have the same chromosome number. So we call this mitotic division as a equational division. So from the given options, option B is the correct answer. Question number 4. Prophase is characterized by the given options: A. Condensation of chromosomal material, B. Centrioles move towards opposite poles of the cell, C. Initiation of assembly of mitotic spindle, and D. All. So here, if you take mitosis, in mitosis, or we can say the mitotic division phase, so there are 4 stages are there: prophase, metaphase, anaphase, and telophase. After interphase, the cell will enters into the division phase, that to be into the prophase, where during the interphase the chromosomes will spread inside the nucleus, and they are slender and thin structures. And when the chromosomes, when the cell enters into prophase, the condensation of the chromosomes will occur. So condensation means the coiling of the chromosomes or chromatin material will occur, so that it is converted into short and rod shaped structure called as chromosomes. So this is one thing. And at the same time, if you take the animal cell, so centrioles are the structures which are helping in the formation of a mitotic spindle or spindle apparatus. So these centrioles will move towards the poles. During this prophase. And not only that thing, when the centrioles will move towards opposite poles, the centrioles will form into an spindle like structure. They are, we can say, they will assemble, or initiation of the assembly of these centrioles will occur, leading to the initiation of the mitotic spindle. Initiation of formation of mitotic spindle. So all these changes are taking place during the prophase. So from the given options, option D is the correct answer. Question number 5. Kinetochores are disc-shaped structures associated with. The given options are A. Telomeres, B. Centromeres, C. Chromomeres, and D. Secondary constriction. So this question is related to kinetochores. Kinetochores are the disc-shaped structures which are associated with the structure of a chromosome. If you consider this is the structure of the chromosome, the 2 chromatids which are present in the chromosome, and inside this chromatid, these are 2 chromatids, one and the second chromatid, inside which the coiling of the DNA structure will be present like this. Like this they will be associated, and in the central part we can see some disc shaped structures like this. These disc shaped structures are called as a kinetochore. And here these are the structures where the bending of the chromosome is possible, and we can call this structure where the kinetochores are present as a primary constriction, or the other name for primary constriction is centromere. And due to presence of this kinetochores, the spindle fibers will come and attach to the primary constriction region, which will help in the arrangement of the chromosomes on the equatorial region of the cell during metaphase. So kinetochores are the structures which are associated with centromeres. So from the given options, option B is the correct answer. Question number 6. Chromatids of each chromosome is separated during. The given options: A. Anaphase, B. Anaphase 1, C. Anaphase 2, and D. A and B. So here separation of the chromatids. So separation of chromatids will occur during both mitosis and also during meiosis. If you consider the mitosis, and this is a cell where the chromosomes are arranged on the metaphasic plate during metaphase stage. So this is a bipolar spindle. This is a diagram related to the metaphase actually. And after metaphase, what happens is the spindle fibers will contract. That is, during the anaphase, this spindle will contract and this spindle will contract, so that these spindle fibers will try to pull the chromosomes towards this side, and these spindle fibers will try to pull the chromosomes towards this side. So because of that pressure, what happens is the chromosomes will split at the center. That is, the centromere will split, and when the centromere will split, the chromatids are separated. So like this, we can see these chromatids are moving towards the opposite pole because of the division of the centromere during anaphase stage. Like this. So the chromatid have been split during the anaphase, and the chromatids are moving towards the opposite pole. So that is, separation of the chromatids will occur during anaphase of mitosis. And when it comes to the meiotic division, so here in meiosis, what happens is meiosis 1 and meiosis 2, two stages are present, and during meiosis 1, the genomes will be separated, or the genomes become half. The diploid genomes will become haploid, whereas during the anaphase 2, the chromatid will split as like here. So let us see that one in the form of a diagram. During meiosis, anaphase 2, the stage where the two chromatids will be, or actually here during metaphase 2, we can see this type of diagram where chromatids are arranged on the metaphasic plate like this. Like this, and here two bipolar spindle, one and the second bipolar spindle here. Two bipolar spindles are present. The same thing is happening as like that in the anaphase of mitosis, where during anaphase 2, during this stage, the spindles, this spindle will try to pull the chromosome towards this side, and this spindle will try to pull the chromosomes towards this side. Because of this one, the chromatid will split, and the two chromatids will get separated from each other like this. So during anaphase 2 of meiosis, the chromosomes will get separated. So from the given options, option D is the correct answer. Question number 7. The following character is not observed during anaphase. The given options are: A. Centromere splits. B. Chromatids move towards equator. C. Chromatids move towards poles. D. Centromeres of chromatids are oriented towards poles. So here during anaphase of mitosis, what happens is the chromosomes will split here. The chromosomes will split at chromatid region, and those chromatids, due to the bipolar spindle, these chromatids will move towards opposite sides. Consider this is one chromatid that have been separated out, and these are the chromatids which have been moving towards this pole, and one more, the pair. Actually, these two are the chromatids of one chromosome. These two are the chromatids of one chromosome, and these two are the chromatids of one chromosome. So, as a centromere will split, during the anaphase, and after splitting, the separator chromatids will move towards the opposite poles. Move towards opposite poles due to the contraction of the spindle fibers. So, here from the given options, one option is given that the chromatids will move towards the equator. Equator means they have to move towards the center of the cell, but the chromosomes, the chromatids which have been separated, are not moving towards the equator but moving towards the poles, that to be towards the opposite poles. So, from the given options, which one is not observed during anaphase means chromatids move towards the equator is not being observed during the anaphase of mitosis. So, from the given options, option B is the correct answer. Question number 8. Precursor of cell wall is, the given options are A. middle lamellum, B. cell plate, C. mitotic spindle, and D. metaphase plate. So, here during division phase, two stages are there. One is called as karyokinesis, and second one is called as cytokinesis. Karyokinesis is the division of nucleus, and when the nucleus is divided, it forms into two daughter nuclei. The two daughter nuclei will be retained inside one cell. Likewise, this is one daughter nuclei and this is the other daughter nuclei. And at the end of this karyokinesis, what happens is the nucleolus will be reorganized or reformed. This is the nucleolus which have been reformed in the nucleus, daughter nucleus, and these are the chromosomes. And after the completion, this is a stage where after completion of the karyokinesis, then what happens is the mitotic spindles which have remained, that is remnants of mitotic spindle, will get organized in the center of the cell, that is they come to the equator, and they are converted into a structure called as phragmoplast. And it is a swollen like structure. Later what happens is the Golgi vesicles will come to this phragmoplast region, and they will burst and release a pectinous material which is present inside them, and these gaps which have been there will be gradually filled up. So, this structure is called as a cell plate, and the cell plate formation, that is the inclusion of these Golgi vesicles will be taking place from center to the periphery. So, we call this as a centrifugal growth. That is, cell plate formation will always be in the centrifugal manner. That is, it will start from the center and move towards the opposite pole, and they will touch the parental wall on both the sides. And after this, the cell, whatever the cell plate that have been formed, will undergo some physical and chemical changes, or physical and chemical modifications will occur. Because of this one, calcium pectate and magnesium pectate will get accumulated. Then when calcium pectate and magnesium pectate is accumulated, we call that part as a middle lamellum, and middle lamellum is the first wall of the cell wall. That means the cell wall formation will start with the middle lamellum, and the precursor to that one, what is the structure that have been formed, is a cell plate. The precursor of the cell wall is a cell plate. So from the given options, option B is the correct answer. Question number 9. Restoring of haploid phase in the life cycle of sexually reproducing organisms takes place by. The given options are A. Amitosis, B. Mitosis, C. Binary fission, and D. Meiosis. Here, restoration of haploid phase. Here, this is a process where can be seen in the sexually reproducing organisms, particularly in the lower organisms. Here the option that have been given is amitosis. Amitosis is a process which occur in prokaryotic organisms, and this is the type of division where the spindle fiber formation does not occur. And one more thing, that it occurs only in the haploid type of organisms, and the restoration is not there. And second one is the mitosis. Here this occurs in haploid cells, where these cells will form daughter cells which are also haploid in condition. And if it occurs in the diploid cells, the mitosis, if it occurs in the diploid cells, the daughter cells will also be in the diploid condition. So whatever the ploidy that is present in the parental cell, the same type of ploidy will be maintained in the daughter cells also. Whereas in the binary fission, the same thing, that is whatever the chromosome number that is present in the parental cell will be maintained in the, will be transmitted to the daughter cells in an equal manner. So here equal division of the cells will occur in unicellular organisms like in bacteria. And when it comes to meiosis, meiosis is a process, so we can call this as a reduction division, where the chromosome number is reduced to half. For example, if a diploid cell or diploid parental cell, if it undergoes meiosis, it will form 4 haploid daughter cells. That means whatever the ploidy that have been transmitted will be half the number of chromosomes which are present in the parental cell. That is because of this one, the restoration of the haploid phase is possible by this meiotic division. So from the given options, option D is the correct answer. The following stage is incorrect with reference to meiosis. Options are A. Nucleus divide twice, B. Chromosomes divide twice, C. Chromosomes divide once, and D. Centromere divides once. So here statement is related to the meiosis division. So here when a cell undergoes meiosis, it will undergo meiotic division, where 2 stages are there. One is called as meiosis 1 and other is called as meiosis 2. So in meiosis 1, so what happens is first the nucleus will divide to form 2 daughter nuclei, and the 2 daughter nuclei will enter into this meiosis 2, and further it will be converted into 4 daughter nuclei. That means here the nucleus is dividing twice. And but what about the division of the chromosomes? Here during meiosis 1, what happens is only the genomes separation will be taking place. Whereas here in the meiosis 2, during anaphase 2, the chromosomes will divide, that is splitting of the centromere will occur, so that the chromosomes will be moving towards the opposite pole. So we can say the daughter chromosomes will be formed as a result of the division of the chromosomes during anaphase 2. Whereas in anaphase 1 of meiosis, only the genome separation will occur. We can say that if a cell is diploid 1, so the 2n will be converted into n condition during anaphase. So the chromosomes are not divided, so that means the chromosomes will divide only once during the meiotic phase. And of course, when the chromosomes are dividing, as I said that the chromosomes will divide only because of the division of the centromere. Then only the chromatids will get separated from each other, and those chromatids, or we can call them as daughter chromosomes, will move towards two opposite poles. So the centromere will divide only once. So from the given options, we can say that chromosomes divide twice is the wrong option, and the correct answer is option B. Question number 11. Synapsis involves pairing of the given options: A. Homologous chromosomes of same parent, B. Homologous chromosomes of different parent, C. Non-homologous chromosomes of same parent, and D. Non-homologous chromosomes of different parent. So here synapsis is a process of pairing of the homologous chromosomes will takes place. So here what do you mean by these homologous chromosomes? Homologous chromosomes are those which are coming from different parents. That is, the origin is from the different parents. One is called as maternal parent and other is called as a paternal parent, or we can call them as a maternal chromosome and other as a paternal chromosome. So here these two will come and get attracted towards each other, and they form a pair. So here we are calling the chromosomes which are coming from the different parents as homologous chromosomes, because the chromosomes which are similar in all aspects except in their origin are called as a homologous chromosomes. Here, as I said, that one is coming from maternal, one is coming from paternal. So the origin is different, but exactly they are having a similarity in all other aspects. And here during the synapsis, these homologous chromosomes which are coming from the different parents will undergo pairing process. So we call this process as a synapsis. So from the given options, option B is the correct answer. Question number 12. The following is incorrect with regard to meiosis. The given options are: A. Nuclear envelope disappears, B. Nuclear envelope reappears twice, C. DNA content of daughter cells is reduced to 1/4 of parental cell, and D. DNA duplicate once. So here during meiosis, as we had already discussed, meiosis will occur in the diploid cells where the chromosome number will get reduced to half, and in the course of all the changes which are taking place during meiosis, the nuclear envelope will disappears. Generally the nuclear envelope will disappears during prophase, both during prophase 1 and also during the prophase 2, and the nuclear envelope will reappears also. Reappears at the end of karyokinesis, that is during telophase 1 once, and during telophase 2 second time. So twice the process will be taking place. And when a daughter cell is formed, so what happens is when a diploid parent cell divide, so the chromosome number will be reduced to half as we have said, but the thing is one parent cell will be converted into 4 daughter cells, and whatever the DNA content that is present in this in the parent cell will becomes 1/4 in the daughter cells. And the duplication of DNA. The duplication of DNA will occur during the interphase. For meiotic phase, so here the duplication of DNA will occur during the interphase. So from the given options, option D is the correct answer. Question number 13. Identify the correct statement. Option A. Each metaphasic chromosome shows 4 DNA molecules. B. Each metaphasic chromatid shows 1 DNA molecule. C. Each chromatid shows 2 DNA molecules in all stages of meiosis. D. Early anaphasic chromosome shows 1 DNA but 2 kinetochore discs. So here option A, if you see, the metaphasic chromosome shows 4 chromatids. Actually, the metaphasic chromosome will appears to be like this: one chromatid with one centromere with the 2 chromatids can be seen, and each chromatid will contains 1 DNA molecule. So the number of DNA that the metaphasic chromosomes will have only 2 DNA molecules. And second option, each metaphasic chromatid shows 1 DNA. Here this is correct. Each metaphasic chromatid will have 1 DNA molecule. Like I said that this is a chromosome of metaphase, and this is one chromatid and this is second chromatid. Each chromatid will have only 1 DNA molecule. This is 1 DNA present in one chromatid, and this is a one other DNA present in the second chromatid. So this one is the correct one. And third option, if you see, each chromatid shows 2 DNA molecules in all the stages of meiosis. Of course, here depending upon the condition, so some stages will show 1 chromatid and some stages of chromosomes will show 2 chromosomes or 2 DNA molecules. So that statement is wrong. And the fourth statement, early anaphase chromosomes show DNA but 2 kinetochore discs. So in the early anaphase stage, only 1 kinetochore disc is present in the chromosome. So that statement is also incorrect. So from the given options, option B is the correct answer. Question number 14. Which one is not possible due to mitosis? A. Wound healing. B. Regeneration. C. Grafting. And D. Reduction of chromosome number. So here, if you see the significance of mitosis, so wherever the wound occurs or whenever the need arises in the plant cell, the differentiation or de-differentiation will occur, where the mature cells will regain that capacity of division, and they will form the new cells in the wounded part. So that is called as wound healing. That is because of the meristematic activity of the cells. So in the meristematic cells, as we know that the type of division will be mitosis. So wound healing is possible by mitosis. And second one is regeneration of the lost parts. So here regeneration is also done by it is a process which occurs in the vegetative parts of the plant body. In the vegetative parts, the type of division will be mitosis. So this is also possible. And third one is grafting. Grafting is the technique of joining 2 different plants, that is parts of 2 different plants have been taken and joined by a process called as grafting. There also the division of the cells is the basic principle which is involved in there. So the division of the cells is by mitosis. So grafting is also been possible by mitosis. And the fourth one, reduction of chromosome number. So here the reduction of chromosome number, actually the mitosis does not lead to the reduction of chromosome number, because whatever the chromosome number that is there in the parental cell, the same number of chromosomes will be maintained in the daughter cells. So whereas reduction of chromosome number is the significant character related to meiosis. So from the given options, option D is the correct answer.